Physiology of flowering. Based on the photoperiod requirement, we have categorized plants into three main types, like long day requiring plants, short day requirements having plants for flowering, and the plants those which don't have any effect whether the duration of light is more than critical value or less than value. It affects the flowering. Instead, in any photoperiod condition throughout the year, day neutral plants they can flower. Now, photoperiodic induction. So based on experimental findings, it is observed that continuous favorable photoperiod till blossoming is not essential, but short and appropriate photoperiod exposure is required for production of flowers. This kind of photoperiod influence persists even when plant is preceded and followed by unfavorable photoperiods. This phenomenon of producing photoperiodic influence is known as photoperiodic induction. Also, it is called as a photoinduction. Salisbury, an American plant physiologist, proved that induction of flowering depends on intensity of stimulus. For flowering, plant may require one to many inductive cycles of photoperiod, which are known as photoinductive cycles. So different plants require different photoinductive cycles for flowering. Some of the examples, in case of xanthium, only one photoinductive cycle is enough for formation of floral primordia. While in case of salvia, this plant requires 17 photoinductive cycles for flowering. Plantago is the another plant which needs 25 such photoinductive cycles for inducing flowering. Besides length of minimum photoperiod, a minimum vegetative growth is also essential for flowering. Photoperiodic induction can be defined as the persistence of photoperiodic effect for flowering in plants. Now, side of photoinductive perception. Scientists Nott and Chailakan, they confirm that photoinduction is perceived by leaves. They worked on xanthium or cocklebur plant, where they have isolated the leaves and a single leaf was kept intact, and it was capable to receive photoinductive influence. So this was their observation. The sensitivity of perceiving influence increases with the growth of leaf till full expansion and decreases in the older leaves. It means process depends on age of the plant even. Now, how transmission of stimulus takes place. While studying photoperiodic influence, careful observations reveals that photoperiod influence is induced in leaves. But flowers are formed in the specialized region of the plant. Then one must conclude that stimulus must travel from the leaves to the flower forming region. And experimental findings also support the view where scientists came to know that this stimulus moves through phloem, and this movement is independent to the transport of photosynthesis. What is the nature of stimulus? So transmission of stimulus indicates that it is in the form of some chemical. Scientist Chalak Khan named this flower inducing chemical as a florigen. And this thing was confirmed while doing the grafting experiment on the Maryland mammoth variety of tobacco, in which case a branch of leaf of plant which has received proper photoperiodic induction could induce flowering on grafting in the plant exposed to unfavorable photoperiods. So this is shown over here in this figure where three sets have been arranged in A, B and C. A, B and C. These three sets of pots potted with the plants. So one plant is of tobacco, Maryland mammoth variety, while the another plant is Hyoscea messenger, the another one. So these two plants in B set, they have grafted these two plants, and later what happens in C, they have shown actually A plant here received proper photoperiod, but B was not. And when such two plants they have grafted, so after grafting, later they came to know both the plants they flower. Both the plants they flower. And it is B plant, though it is not having proper photoperiod, then also it got flowers. It means from A plant the stimulus or photoperiodic induction received plant which was having some chemical now is transferred through the graft to the plant B, and that's why B plant is able to flower here. So this is how the experiment performed by the scientist Chalak Khan gives support that the stimulus is in the form of chemical, and he has given name to it as a florigen. So this florigen through graft, it grows from produced in the A plant, gets transferred to the B plant, and B plant is now able to flower. Both the plants they flowers. So this is the experiment which gives us idea that the nature of stimulus is in the form of chemical. Now photoperiodism and quality of light. So there is a relation. Green color of visible spectrum is ineffective in inducing flowering, while blue color induces poor flowering. And wavelengths ranging from 580 nanometer to 680 nanometer in the red portion of visible spectrum are found most effective for inducing flowering in both short day and long day plants. Now phytochrome and flowering. Experimental findings shows that brief exposure with red light during critical dark period inhibits flowering in short day plants. This inhibition or inhibitory effect can be reversed by subsequent exposure to far red light. Prolonged light period is essential for flowering in case of long day plants. Thus, inhibition of flowering in short day plants and stimulation in long day plants involves the operation of a proteinaceous pigment. And name of that proteinaceous pigment is phytochrome. So what is this phytochrome? Actually, it is a photoperiod stimulus receiving pigment. Phytochrome is photoperiod stimulus receiving pigment. And it has some characteristic features. It is proteinaceous pigment. It is universal in distribution in green plants. It has two forms called as PR and PFR. And these two forms, they are interconvertible. PR means red light absorbing form of phytochrome. It absorbs maximum light in red region of the spectrum at 660 nanometer wavelength. And other PFR means far red light absorbing pigment. This PFR form absorbs maximum far red light region at 730 nanometer wavelength. Under long day photoperiods, what happens? P730 is retained for longer time under long day periods. P730 form of phytochrome is retained for longer time and is responsible for stimulating flowers in long day plants. And at the same time, it suppresses flowering in short day plants. Now under short day photoperiods, another form of phytochrome, that is P660 nanometer, is retained for longer time, which stimulates flowering in short day plants and at the same time inhibits flowering in long day plants. Thus, it is presumed that these two forms, PFR and PR, of phytochrome regulate formation of substance or hormone, which on other hand influence flowering. So here PR PR can get converted into PFR under red light condition, and PFR again gets converted into PR under far red light condition. PFR form of phytochrome shows another type of conversion. PFR spontaneously becomes converted into PR in dark, which is called photoconversion. This conversion is very slow and takes few hours, and process is dependent on temperature, and therefore it is also called as a thermal conversion. PFR form of pigment gradually changes into PR form in dark. During daytime, PFR accumulates in the plant and inhibits flowering in short day plant, while it stimulates flowering in long day plants. In critical dark period in short day plants, PFR is converted into PR form, resulting into flowering. Phytochromes and flower initiation. Actually, flower initiation is very important aspect while considering the agricultural products, where flower production and therefore fruit formation and seed formation, these things occur, and for getting maximum yield, the flower initiation is must. So for this purpose, the phenomena like photoperiodism, vernalization, also carbon nitrogen ratio and rate of photosynthesis, these all phenomena play a crucial role in induction of flowering. Here we will discuss the role of phytochromes in flowering. So some of the oxygens like NAA or IAA, indole acetic acid, can promote flowering in plants. For example, plants belonging to Bromeliaceae. Gibberellins are also there. They play important role in induction of flowering. Even BZA treatment, blue-green algae treatment, can be a substitute for photoinduction or a specific cold requirement. Gibberellic acid can induce flowering even in absence of photoperiodism and vernalization. Whether plant get proper photoperiod or whether it get proper temperature or not, that do not affect the flowering if one apply gibberellic acid to the plant. Thus we can say that gibberellic acid can induce flowering in absence of photoperiodism and vernalization even. Gibberellic acid can induce flowering in long day plants or even short day plants, even though there is no proper photoperiod received by the plant. Gibberellic acid is able to induce flowering in some biennial plants. One of the gaseous hormone, ethylene, also induces flowering in plants. For example, pineapple. Photoperiodism, a phenomenon, is of importance, and how one can apply it for the commercial farming. So those applications we will see here. So this photoperiodism phenomenon is of great importance in case of hybridization experiments. So for flowering, the researcher will not depend on the season. In any season he can get flowers, and hybridization experiments or work can be accelerated. Isolation and characterization of hormone fluorescein is of economic importance. Photoperiodism is good example of physiological preconditioning, where external factor induces some physiological changes in plants. Some more uses of photoperiodism. Winter varieties of rice flower. Earlier under short days, whereas summer varieties under short days should delay in flowering and prevent earliness due to high temperature treatment. Long photoperiod increases flowering in Indian wheats, and these plants require low temperature at early stages. Normal day length has great significance in productivity of plants.